Welcome to the Axial Podcast. Axial is an early stage investment firm based in San Francisco. We partner with great founders and inventors investing in early stage life science companies often when they are no more than an idea. Axial is fanatical about helping the rare inventor who is compelled to build their own enduring business. You are? Okay, Basem, let me make you a moderator.
All
Yes,
right, sounds
I'm
good.
sure. Hey, Basem, how are you? How's your day been?
Hello.
Can you hear me?
Yeah, hello.
Okay, cool. Well, let me do the introductions and then we can kind of get to it.
Okay.
Thanks for everyone for showing up today. I have a really awesome guest, Basem Al-Shayeb from the University of California, Berkeley, and he's a graduate student in the Jill Banfield lab and also Jennifer Doudna's lab jointly, if I remember correctly. Cool.
Yes, that's correct.
And so Basem is the lead author, and he's wrapping up his thesis. I just got a text from my friend Rishi that he's happy that you showed up and it works this time. But Basem was the lead author on some really awesome preprint discovery, I think called Borgs, which we're going to discuss today. And he's wrapping up his thesis. And so, really excited to talk to you and learn more about Borgs and kind of the future directions and the whole story too. So maybe to start off, maybe a quick introduction on you and your career, and then we can kind of dig in.
Yeah, and yeah, thanks for having me. I'm really excited to be talking about this work. So I'm currently a fifth year NSF fellow at the UC Berkeley, conducting research with Jill Banfield and Jennifer Doudna. And the sort of theme of my thesis so far has been the discovery of new types of mobile elements, so new groups of viruses, things like Borgs, and also sort of developing new genome editing tools that could have applications in human health or agriculture or even diagnostics. So really, for me, the most exciting part has been the sort of basic science discovery through sort of curiosity-driven exploration of the world that we live in, and then evaluating how those discoveries could be used as useful tools for society. Um, so yeah, the latest of that has been this Borg paper that was really part of work that we stumbled upon um, while conducting research on these giant phages and different CRISPR systems.
Cool. I mean, maybe to like set the stage first. I think it's always important to not only understand the science, but the person who did the science. And so, what kind of got you interested in biology in the first place?
Yeah, so for me, initially, I've always been fascinated with how different things in biology work. Obviously, I'm a big fan of tech and computational work, which is the basis of a lot of the work that I do. But at the end of the day, nature has been um, to me the most fascinating tech, just because it's evolved for billions of years in ways that we could never have imagined. And for me, it's really rewarding to discover mechanistically how different things work in the natural world. So actually, my physics teacher in high school was really sad about me not going into physics and engineering and going instead into biology, because I really enjoyed obviously physics and engineering, and that's why I have still stuck with synthetic biology. But biology still has always been my passion. Um, and so I did my undergrad at University of Minnesota, studying genetics, cell biology, and development, and also a major in microbiology, with a minor in philosophy, looking at philosophy of science and how we interpret different kinds of data. So all of that has sort of culminated in the role that I have now, doing a PhD in microbiology at UC Berkeley, on these different topics.
Cool. It seems like you're just a really well-rounded person. Physics, philosophy, and biology. And did you go to? You have the NASA logo as in your background. Did you kind of go to space camp as a kid, or what was kind of some formative? What was were there any like formative scientific experiences? You mentioned the physics teacher, kind of being a mentor and guiding you. But was there any kind of like formative experience that said, "I'm going to be a scientist"?
I mean, space and astrobiology have also been, big passions of mine. I worked for a little bit, during undergrad and after undergrad for a bit at NASA Ames Research Center with, their space biosciences division. So I worked in April Ronka's lab, where, they study sort of the theme is sort of future generations in space. So how, space flight conditions could affect, future generations, and for example, pregnancy under spaceflight conditions, pregnancy under, hypergravity or microgravity conditions, how those things can affect, um, if we end up going to space at some point, how, we would essentially form a colony, how those things would affect, future generations biologically. So that research, there's a lot of, qPCR, ELISAs, looking at, different gene expressions and protein production and things like that under, spaceflight conditions. And I'm still, in touch with the different scientists that work in that division, looking at future collaborations and stuff. So it's definitely still a um, big passion of mine, space biosciences.
Cool. That's really my old mentor Gary. He had this project. I don't know if he's done it yet, but he wanted to send like a nanopore to Mars just to see if there's anything to measure. I don't know if that's happened, but yeah, I think totally agree. I think understanding the interplay between biology and space is a pretty interesting opportunity. And so maybe to shift gears a bit and kind of go towards your experience at UC Berkeley and really understand the story of not only the story of how you discovered Borgs and then the meaning of it. Did you ever overlap with like Brian Thomas or Dudu Bernstein? Any chance? Any sort of
A couple of months at the beginning of my PhD. Yeah,
cool. Yeah, Dudu's a great guy. I think he's at the Weizmann now, or I think he's somewhere in Israel. But he's a great guy.
yeah, he is.
Cool. And so maybe for the Borgs, what was the kind of the so you enter the lab. Was your project? What was your project? What was the kind of the initial hypothesis that like you set upon, and that ultimately led to Borgs?
Well, initially I was studying giant phages. So, giant viruses that infect bacteria. And from a specific site where, it was really a spontaneous decision to sample initially. So we were just in this area that was near, Jill's house. And we thought, why not take some samples to sequence and look at in the lab? And that was sort of a very wet bog area, in a forest that, um, we didn't really think twice about. And then once we found that with very deep sequencing that they do have these giant phages, we decided, the next year I'm going to go back and I'm going to collect a lot more samples, try and study these phages a bit more, and, understand really more of the biology that's going on in that environment. And what we found was these giant sequences from the sort of, computational assemblies of genomes in that sample are these giant sequences that looked more archaeal than bacterial. Um, and for the first few months, we just called it the archaeal mystery. As I finished the sort of giant phage work, um, this was something that I was still trying to figure out: is this a virus? Is this a plasmid? Um, until I found sort of the complete genome, that was something that was just like a weird sort of artifact, or something that I, you know, shelved for a bit until I work on clearer research directions. And then we ended up with the complete sequence, with more samples from the site, and it looked, um, as we saw, very archaeal, and that archaea are arguably the most understudied domain of life compared to bacteria and eukaryotes. So really, there are quite a lot of mysteries to still learn about them. Um, I remember I have a folder of most mind-blowing science videos that I've seen, and one of those videos is a video of an archaeal cell that instead of dividing into two cells, it divides into three cells. So it's this like triangular cell that looks like a Dorito, and it divides into three daughter cells, which blew my mind.
That's pretty
Uh,
crazy. That's pretty crazy.
yeah, it's in a folder along with like phage nuclei and different mind blowing science things. But these archaea are very obviously understudied, and there's quite a lot there that still needs exploration. There's not a lot of like model systems that are studied in labs, and obviously a lot of them are anaerobic. So that was something that I really aimed for in the second sampling that we did. Was we got these tools to dig really deep holes in the ground, and I used that to make a hole and then stick my arm with gloves and stuff all the way as far as I could reach with my arm, which was a lot colder than I expected. So like for me, I'm from Cairo, from a city, so doing these sort of sampling trips in greenery and forests was really exciting for me. And I dug my hand as deep as possible to get these really anaerobic samples with very little oxygen, that were rich in different archaea. Um, and these archaea specifically that we found to be the Borg hosts, that we call methanoperidins, these were only recently discovered, and they are known to extract methane from the environment and metabolize it. So as soon as we found this element that sort of had genes similar to the methanoperidins, we thought, okay, maybe this is like a sibling lineage to the methanoperidins because it's linear, and it seemed to have these inverted repeats at the ends. So they didn't look like, they didn't have any viral structural genes. They didn't have the canonical plasmid replication genes. We thought maybe this is a sibling lineage to methanoperidins, especially that it had a ribosomal protein that obviously is used to make proteins in the cell. That ribosomal protein was really similar to the one from methanoperidins, along with other genes that were really similar as well. But that was we only found one or two ribosomal proteins in the complete genomes, which meant that it can't survive by itself as a sibling lineage. And a lot of other genes that you would consider essential for life were also not there. So through a process of elimination, essentially, we um, decided that this was a extrachromosomal element of some kind, and then started digging more into what other genes are encoded there. Um, and there's this rich diversity of these methane oxidizing organisms like methanoperidins, but right now we don't know fully how to grow them in the lab to study them in depth. So, but obviously there's a pressing need for us to study this biology, since methane is a greenhouse gas that's 30 times more potent than carbon dioxide. So it obviously affects climate change in a drastic way. And this is, methane that's produced is through, transport of coal, of natural gas, of oil. There's obviously a lot from livestock and agriculture and from landfills. But right now, all the focus is on capturing carbon dioxide. But even the biggest facilities that I know of that are in Iceland to capture carbon dioxide, they do. I think it was like 4,000 tons of carbon dioxide per year to capture, but that negates maybe 3 or 4 seconds of the global emissions. So obviously, there's a pressing need to understand how something like methane is actually removed from the environment. And that's mostly currently done by microbes that we don't really know a lot about. Um, so for us,
Exactly.
instead of,
Yep.
sorry?
Exactly. I think like a big part of it is biogeochemical cycles aren't really well understood, and this is what made your research really exciting because then begin to understand how do we integrate microbial cycles with like these kind of carbon models of the Earth. But maybe to back it up, could you maybe describe Borgs? This kind of these large elements. They have these direct repeats. And also, who named who? It's a great name. Who came up with the name Borg?
Yeah, so like I said at the beginning, we just called it the Archeal mystery, and it stayed that way for a while. And then once Jill was telling her son actually about this research, because we found that these Borgs, they're linear elements. They're up to a megabase in size, which is a third of the size of their host chromosomes, which is it's pretty massive. It's bigger than any of the giant phages that we described. It's bigger than a lot of small bacteria. So this was, really fascinating to us, but also, like I mentioned, it had a lot of genes from the host. So this transfer of genes through horizontal gene transfer from the host, Jill was describing to her son, and he just said, "Oh, so it's like Borgs from Star Trek," because in Star Trek, Borgs are this cybernetic group that needs to assimilate technology from other species, from like a sort of host to keep up their collective. And so we found this to be a really nice analogy with studying these linear genetic elements, because the majority of the genes that are encoded on the Borg DNA, up to 80%, are currently unknown. We don't know, like it's not similar to anything that we found outside the Borgs, and obviously nothing characterized in the lab. And so we figured that this was a chromosomal element, extrachromosomal element of some kind in the methanoperidine cells that seems to assimilate a lot of genes from the methanoperidines. And that includes, for example, the methane oxidation group of a cluster of genes. Um, so that's how we came up with the Borg.
That's a great story, I think. And so you spent a lot. I mean, I'm sure it was a lot of work, just like legwork, to get the samples and then analyze them and then reconstruct these elements and then really then hone in on saying these aren't megaplasmids, these aren't from phages. This is kind of something unique. How did you go about? You touched upon this in the preprint around their potential function, and you have like a nice little figure about trying to like figure out what they actually do in terms of augmenting methane oxidation. But you discovered Borgs. How did you go about then trying to figure out what they do, or at least hypothesize what they could do in the environment?
Yeah, so once we had the complete genomes, we sort of went through and annotated everything as either, something that was new and uncharacterized, or as, the 20% of genes that had similarities to existing proteins in, the NCBI database and other databases. We went through those annotations to try and figure out which processes those genes are part of. And methane oxidation is one thing that we touched on. There was also clusters of genes for nitrogen fixation. So with something like nitrogen fixation, it's very important in these soils that we found them in, and these are also found in um, different rice paddies and in many different environments. So it's nitrogen fixation is very important agriculturally since plants require nitrogen compounds for growth. And they essentially have transferred this machinery to their own genome, but also different genes, for example, for transferring electrons outside the cell. So the way that they respire is by transferring their electrons to another compound, for example, different metals, something like methane, instead of oxygen. So that's a sort of long way of saying that they breathe other things than oxygen. And this was something that, I worked in a lab in undergrad with Jeff Gralnick, who studies this pretty extensively. So it was really rewarding for me to sort of come full circle and touch upon those topics of transferring electrons outside the cell. That, potentially this could be something that can be used to generate electricity, since electricity, all it is transfer of electrons, whether it's through a wire to an electrode, and so on. So these likely enhance their host's ability to metabolize different compounds, methane and other metals. So those are some features that we were most excited about. But they also have different defense systems. For example, they have their own CRISPRs. So analogous to the discoveries that we made in phages in my previous papers, we found that they have CRISPR systems that target also other mobile elements that are integrated in the host genome. So clearly they are competing maybe for the host, or augmenting the host's defenses against different mobile elements that would inhabit the same cell. And the other really fascinating thing that we saw was that different Borgs could also exchange genetic material with each other, and not just with the host. So there are, blocks of 11,000 base pairs, for example, that are 100% identical in different Borgs, which implies that those two Borgs probably were in the same host cell at some point, and there was some recombination that happened that transferred that piece of DNA. So, and yeah, those are just the things that we already know the functions of. And there's, 80% of the genome is still unknown. So there's a lot still to learn from them.
Really cool. When I read the paper, I'm like, my gosh, Basem has a decade's worth of work ahead of him here. Like, you can do so much. There's just so much to explore. I remember, I think Jill's lab published a paper in 2016 discovering the first CRISPR system in archaea. And so I think you're spot on. Archaea is so underexplored branch of life, and there's probably so much cool biology and tools to be discovered there. And so then you made
Yeah,
this.
I mean, even the first CRISPR, when CRISPR was first described back in the 80s, a lot of that was from archaea.
Oh, really? Interesting. I thought the first CRISPR systems were from yogurt, from Dannon. Or maybe
Oh no, so
I
that
miss.
was the first indication of what CRISPR could be doing. But the very first, when they came up with the word CRISPR, the discovery of the repeats was back in the 80s. That included archaea.
Wow, interesting. That's really cool. I did not know that. I need to go read up some history. But so you made this discovery on Borgs, and then you did a lot of hard work to infer their function, and now you and the whole lab probably hard at work right now trying to learn more about these Borgs. But the really exciting part, and you alluded to this, is how they can modulate greenhouse gases, in particular methane, and microbes play a massive role in carbon dioxide flux. I think it's involved in at least half. Maybe you can correct me on this, of fixation of carbon dioxide in our environment. But microbes play a really pivotal role in carbon cycles, and so maybe you should touch upon maybe the interplay between Borgs, biogeochemical cycles, and climate change. It's a pretty large space, but do you have any kind of ideas around the potential impact of Borgs on the climate?
Yeah, I mean, for right now, we're still even just trying to understand what kind of diversity is there for Borgs, where are they found, why are there, for example, specifically some methanoparidins that have Borgs and some that don't. So obviously, like I said, methanoparidins was very recently described as a species of archaea. But going through that data, we couldn't find any Borgs in their data. So why is it that they exist on the methanoparidins that we found, but not in somewhere else like Australia? Um, so that there's still a lot to learn with their impact and their distribution across the world. And also, this was just the first demonstration of the methane oxidation machinery being able to transfer to mobile elements, and with that also implying transfer across lineages. So that's something that would be really interesting to explore, how much of that is mobile across different lineages of these both archaea and bacteria that tend to metabolize methane. Um, but yeah.
Really interesting. Yeah, I think is I'm really horrible at pronouncing like species names. That's what C. elegans is such a nice. It's so easy, right? Drosophila, but Methanoparidins. Okay, I got it. Is it a heterotroph or is it something else?
Um, I think it's I think it is a heterotroph.
Okay.
I don't think there is any evidence of it fixing its own carbon. Um, but yeah.
Yeah, because my understanding of like how I took this class like over a decade, I think a decade ago, a guy named Peter Gurgis. Like, so he's really he's this
Oh
cool.
yeah.
He's awesome. I think I don't know where he's at. I haven't kept in touch with the guy, but he's just
He's
really.
in at Harvard. Well,
Yeah,
I know because he's another fellow Egyptian.
oh, he is. Oh, cool, man. He's a cool guy. I think he used to work at Monterey and then went to Harvard. But I took a class of his. I was really fascinated with like microbes in the ocean. Right? Isn't that? I love history, and so like I think a very romantic part of biology is like Darwin on the Beagle. Rather than being in lab all day, he could be on the ship. That's much more attractive to me, I think. But and from my understanding of how biogeochemical cycles work, heterotrophs are really involved in oxidation and the production of CO2, and then photoautotrophs are really involved in fixation and maybe production of oxygen. So just something I'm not. I've been out of science for so long, so I'm not on the cutting edge anymore, unfortunately. But and maybe that's maybe there's some sort of Borgs play some element in terms of oxidation, and but to be determined.
Yeah, I
Yes,
mean the methanoparidins I think would be considered like chemolithotrophs. Um, so using like inorganic sources, like um, different metals, for example, for respiration. But obviously, they also use methane.
interesting. Yeah, so much should be done. And I think not only do great science, but probably go on some really cool adventures. Because metagenomics, definitely the cool part about metagenomics is you get really cool data, but then you get to like go on cool trips. Whoever went to that mercury mine in Napa, that must have been kind of scary. To be honest, I would have. I can see why you didn't want to go to that mercury mine. I wouldn't have gone. I would have been terrified. But cool. Maybe then we can kind of then also touch upon the kind of just searching nature for new inventions. And Borgs is something in nature. And in the beginning of the conversation, you touched upon this of being fascinated with nature. And nature invented CRISPR. Jill Banfield, you can argue, invented or was a key inventor of CRISPR, and her data set was what enabled Jennifer to make her awesome discoveries. I think Patrick Shrew just published some cool papers around integrases. But do you have any thoughts around like why nature has been such a great inventor. I think the arms race between phages and microbes seems to be it. But yeah, any thoughts around kind of nature as an inventor?
Yeah, I mean, to me, that has always been my driving force for, research interests. And that's sort of why I decided I wanted to be co-advised between Jill and Jennifer. Is Jill has this innate curiosity towards nature, and sort of the discovery of new basic science. And Jennifer is always asking me, so what? How can you use this for something useful? So that has been a very strong combination in my thesis research. And for me, looking at nature, like arms races have always produced some of our best tools. Even something like antibiotics, that's the arms race of different microbes against each other. The same site where we studied all these giant phages, and also the Borgs, the same site a colleague of mine was using to look at different antibiotics, that the preprint came out for pretty recently as well. Um, and so this you know plot of dirt near Jill's backyard was really the driving force for, or the site for a lot of my thesis research and my colleagues' research. Um, that it just goes to show that everyone has this sort of universe lurking near them, waiting to be explored. And obviously, like as I said, I'm really excited about space exploration and astrobiology and all of that, but there's also this incredible diversity just here near all of us that is just waiting to be explored. And that's what really excites me about microbiology and about science.
Well, it's really powerful, Basem. Like the next great discovery might just be in your backyard. That's a great. That's a
Exactly.
great. That's a great line. Uh, um, and anybody in the audience, if you have any questions or around the topic of science and Borgs, please raise your hand. But maybe to kind of wrap up this conversation, and I know you're doing your thesis, so I don't want to, I don't want to any talk about things that are unpublished. But what kind of new research are you doing? Kind of exploring Borg research, and also maybe going beyond Borgs and trying to find new types of elements in these samples.
Yeah, I mean, of course, a lot of that work is being done, and for me, mobile elements are some of the most fascinating things in biology. Ever since I think Barbara McClintock got a Nobel Prize for transposons, it's always been, elements that do things somewhat differently than life, because they have this selfish need to want to replicate as much as possible and spread everywhere, regardless of what different life forms might have to say about that. And I think that there's a lot of um, untapped tools that can be developed from that. And obviously, that's what I've focused my thesis research on. And that's, a lot more unpublished stuff that I'm hoping to get out soon as well to touch on that.
Cool, man. I'll be if you have transcripts, I'll be your first reader. I love reading paper. I'm very good at reading papers. I'm not so good at doing the science. So I think seriously. I went to grad school at Berkeley. We can go get drinks, but I can tell you my scientific career, and I'm definitely bad at the bench. But yeah, I think I read your preprint at least 10 times. It's not that long, so it's not that impressive. But what are you thinking about after grad school? So you're going to wrap up your thesis. Are you thinking about professorship? Are you thinking about entrepreneurship? You thinking about joining NASA and doing space biology? What kind of what do you think your next steps are?
I mean, I'm keeping doors open right now. All I know is that I'm really excited about creative research to sort of explore the world and come up with sort of translational tools that could be helpful from that. So, whatever context I can do that in is hopefully what I would pursue as my next step. Um, that might mean, academia, but I also know that at least in the entrepreneurial space, it seems people are a lot more receptive to, you know, more high risk, high reward ideas than maybe they used to be. So I'm keeping that option open as well.
Cool. Yeah, we live in the age of the inventor, right? So just if you can invent something new, the money's there. I met Brian Thomas, and he has. Usually, you should start a company. Or you should join Metagenome. That's a really cool company. I met Brian when he started the company, and I did not invest like an idiot. I should have invested, but Brian's done an incredible job. I think maybe serving as an example. I think he was in Jill's lab for a long time, and now he has an incredible business. So I think the future is bright, and I think they have a lot of great options. I think Borgs is a real big breakthrough in kind of connecting microbes with our environment. Understand the mechanism by which like carbon and nitrogen and various other chemicals are kind of recycled, because geochemical cycles are really underappreciated in like climate models. And so if we can kind of hone in on that, we might have different ways to engineer the environment and then solve climate change. But
Yeah,
awesome.
absolutely.
But any final thoughts? I think I had a great time. Great time. I think. But any final thoughts you want to part to everyone?
Um, Maybe just that, none of this work would have been possible without sort of curiosity driven basic research. Like, we didn't really set out to find a tool that might help with climate change, but that sort of came to be from the curiosity driven research that we did from on other things. And the same can be said about CRISPR, for example, where nobody really set out to invent a gene editing tool. That just sort of happened as we studied why these elements have repeats in them, and what are these genes that tend to co-occur with the CRISPR associated proteins that are now Cas proteins. Um, so yeah, there's a lot to be learned from just basic science, I think. If you want to sit and invent something new, it's always a good idea to look at nature and how nature has done it, because that took billions of years of evolution. Um, but yeah,
Totally agree. I think we're on the same page. Well, Basem, I had an awesome time talking. I was really just curious to have a conversation with you since I read your paper a lot, and it was really, I think it's a massive breakthrough, and it's going to unlock a lot of different elements in terms of like biology and climate. But yeah, when you get your thesis done, send me an email. We'll go for a walk. We'll go to Jupiter, or go for some cheese board or something. But good luck on the thesis. I think you probably have a lot of writing ahead of you. Like, how long is your thesis going to be? Is it 100 pages or 300 pages, or how long do you
uh,
think?
I can tell you I've already passed 100.
Jeez Louise, that's a lot. Oh man, how do you edit that thing? Well,
Uh, yeah, that's the struggle.
Well, Bassem, get some sleep, and congratulations on all the success. I'm sure the best is yet to come, though, and so I'm really excited to just send me an email, and we'll go for a nice little walk around Berkeley. But thanks for taking the time to talk to us, and I learned a ton, and I think we all look forward to kind of what you're going to do next, and kind of the impact you're going to have on science and more. So I really appreciate it, everyone, and Bassem, thank you for the time. I really appreciate it.
Yeah, thanks so much for having me. That was fun.
Cool. I'll end the room and we'll talk soon, Basem. We'll have a great day.
Thanks, you too.